Hello, today we are going to know about the vectors. What are vectors? Vectors are DNA molecules which can replicate autonomously in the host cells. They can carry the gene of interest into the host cell. Along with the vectors, inside DNA also replicates. What are the types of vectors? Some vectors are plasmids, bacteriophages, cosmids, phagemids, yeast artificial chromosome vector or yolk vector, bacterial artificial chromosome vector or BAC vector, transposons, they also act as vectors. Agrobacterium tumefaciens acts as a vector for cloning the genes in plants. Retroviruses, shuttle vectors, they are used to clone the genes in animals. These vectors, they differ in the size of the gene that can carry into the host cell. For example, let us take plasmids like a pBR 322. It can carry about a 10 kb length DNA molecule or DNA sequence. Phage vectors can transport 20 kb length sequences. Phagemids, which are hybrid vectors, these hybrid vectors are formed by the phage and plasmid. The phagemids, they can transport 10 to 20 kb DNA lengths. Cosmids can transport 45 kb length sequences. Bacterial artificial chromosome, it can carry about 130 to 150 kb length DNA sequence. Yeast artificial chromosome can transport 1000 to 2000 kb length DNA sequences into the host. And which vector we have to use? That depends upon the size of the insect that we are going to transport and also its application purpose. So by considering these two factors, we have to select the suitable vector. What are the characters of vectors? These vectors should be convenient to carry the gene of interest into the host bacterium. They must be able to replicate in the host bacterium. They should contain unique sites for the action of restriction endonucleases. Those are also called multiple cloning sites. So a vector should have the multiple cloning sites. For the selection purpose or for screening the host cells, they should contain the selectable markers. And these are some main characters of the vectors. Now let us see the one typical vector, what it needs. It needs one ORI site, origin of replication. From this point, the replication of the vector starts. It is also responsible for controlling the copy number of the linked DNA. That is the function of the ORI site. And also, it must have one ROP site or sequence. ROP is also called the repressor of primer, and it codes for the proteins involved in the replication of the plasmid. And this ROP not only codes the proteins and also plays the role of regulating the copy number in a plasmid. So ROP maintains a copy number of the plasmid. Next, selectable markers. These selectable markers may be antibiotic resistant markers. In artificially made vector is the pBR 322. It has two selectable markers. One is ampicillin resistant site and another is tetracycline resistance sequence. So the genes coding for resistant to antibiotics such as tetracycline, ampicillin, chloramphenicol, kanamycin acts as selectable markers. These are the basic things required in a typical vector. And also the vector must have the cloning sites. Cloning sites are the places where the restriction endonucleases can act. Let us see this pBR 322. It has many cloning sites. Here for PST1, one cloning site is there, and PVU1 has a cloning site in ampicillin resistant gene. In the same way, tetracycline resistant sequence has BAMH1, BAMH1 site, and ROP also has one site for the restriction endonuclease, and tetracycline site has one more the cloning site for SAL1. In this way, one plasmid may have many sites for several restriction enzymes, but one plasmid must have only one site for one restriction enzyme. For example, let us take this plasmid. Here, tetracycline resistant marker we are taking. Here BAMH, Bacillus amylophaciens H strain, and the enzyme extracted from it is the 1. And for this, here one cloning site is there. Then it can cut this plasmid into one straight sequence. Suppose if more than one site is there for this BAMH1 on this pBR 322, it will cut that plasmid into many fragments. So those are not convenient for inserting the gene. So a plasmid must have only one site for one enzyme, but a plasmid may contain many cloning sites. These cloning sites are also called multiple cloning sites or MCS. The size of the vector. Generally, we prefer small sized vectors. If the vector size is very big, then it is very difficult to insert into the host cell. There are chances of breakdown of that vector while doing that transformation. So preferably small sized vectors are selected to transport large sized molecules. These are some structural details about the plasmids. One is they must have ORI site, origin of replication, where the replication begins in this plasmid or in this DNA sequence, and one more is there repressor of primer or ROP site, which maintains the copy number, and it must have the selectable markers. One or more selectable markers are preferred, and they must have the many sites for the action of the restriction endonucleases. Those are called cloning sites. And next, the size of the vector should be small. Let us discuss about plasmids in this class. Under this plasmids, we are going to discuss about pBR 322 and PUC 19. What is plasmid? First, let us understand. This is a prokaryotic cell. This prokaryotic cell has many circular DNA. These are not related to the main DNA, chromosomal DNA. So these are called extranuclear DNA rings. These extranuclear DNA rings, DNA sequences present in the prokaryotic cells are called plasmids. Plasmids, they are meant for several purposes. Some plasmids, for example, F plasmid is there, and it is useful for the production of conjugation pili. And some plasmids have antibiotic resistant genes which can withstand the action of the antibiotics. And some plasmids may produce the products which is important for the fermentation of the sugar. Some are useful as heavy metal resistance. In this way, many types of plasmids are present. So anyhow, plasmid is a circular extranuclear DNA is called plasmid, and these plasmids are naturally we can see in the prokaryotic cells. But the plasmid here developed is an artificial one, and this was developed by Bolivar Rodriguez, and so it is called plasmid Bolivar Rodriguez, pBR 322. This is the lab number. The size of this plasmid is about 4.3 kb. It has two sets of antibiotic resistant genes. So here we have to do the two-step selection process. Reasonably have high copy number. About 25 copies they produce, more than 25 copies, pBR 322. And this pBR 322 has two selectable markers. One is the ampicillin resistant site and another is the tetracycline resistant site. And as usual, it has the ORI site and ROP site. On tetracycline resistant site, it has one cloning site for the bomb H1. So how this will transport that gene, let us see. First, the gene of interest will bind to the tetracycline site at BAM H1 site. Now, this formed plasmid with some foreign DNA is called a recombinant plasmid. Now, this recombinant plasmid loses its property to resist to the antibiotic tetracycline because it is disturbed by this. But this recombinant plasmid still shows resistance to ampicillin. Thus, we can produce recombinant DNA or recombinant plasmids. Okay, look here. When the gene of interest is inserted into the plasmid sequence, now it is termed as recombinant plasmid or rDNA plasmid or chimeric DNA also we can call. Chimeric DNA is the DNA with two or more sources of DNA is called chimeric DNA. In Greek mythology, there is one creature is there, name is chimera. Its head is the lion's head, the trunk is of goat, and the tail is the snake. Such an organism is named as chimera. That mythological character is a chimera. And so that chimera has different body parts, different organisms, different animal body parts. The same way here, this recombinant plasmid also has the DNA sequences from different sources. So this recombinant plasmid is also called chimeric plasmid or chimeric DNA. Let us see the transformation. Transporting of the gene of interest into the host cell is called transformation. Or we can say that introducing of plasmid into the host cell is called transformation. Suppose if this plasmid has a foreign DNA, then we call it as a recombinant DNA. If this recombinant DNA is introduced into the host cell, we can say that that host cell is a recombinant transformant. Otherwise not. Okay, let us see this animation. These are the plasmids. One is the recombinant plasmid and another is a non-recombinant plasmid or non-recombinant DNA. And here this recombinant DNA or non-recombinant DNAs are not successful in entering into the host cell. Now this host cell is called non-transformant cell. So what are non-transformant cells? Bacterial cells that do not take foreign DNA are called non-transformants. Suppose if the bacterial cell is successfully received the foreign DNA, then it is called transformant. Transportation of or introducing a foreign DNA into the host cell is called transformation. Now let us take transformants. When foreign DNA enters the cell, then that host cell is called transformant cell. Suppose if a recombinant plasmid enters into the host cell, then we can call it recombinant transformant. Look here, recombinant plasmids are there with the gene of interest inserted into this plasmid. Now this plasmid is successful in entering into the host cell, so it is called recombinant transferment. Now, one more, let us see. These are the plasmids without any insert. So, these are non-recombinant plasmids. When these non-recombinant plasmids entered host cell, that host cell became a transferment, but it received non-recombinant DNA, hence it is called non-recombinant transferment. So, what are non-recombinant plasmids? The plasmids which do not have the insert of foreign DNA are called the non-recombinant plasmid. When the host cell receives the non-recombinant plasmid, then it is called non-recombinant transferment. So that is about the transformation of chimeric DNA or rDNA into the host cell. Now, how to select the recombinant transferments? And for that, the pBR 322 has two selectable markers. One is the ampicillin resistant site, and another is the tetracycline resistant marker. Into this tetracycline resistant marker, we are introducing this gene. Here we inserted this gene into tetracycline resistant marker. When this is inserted into this one, this plasmid loses the property to show resistance to the tetracycline, but it can show resistance to the ampicillin antibiotic. So this is the recombinant plasmid, and this is the non-recombinant plasmid. Now this non-recombinant plasmid we introduced into the cell. Now this is called non-recombinant transferment, and we allowed these non-recombinant transferments to multiply. Now such groups of these non-recombinant host cells, when we keep in ampicillin medium, then these can survive, because non-recombinant plasmid ampicillin resistant gene is not disturbed, so it can survive in this medium. In the same way, the non-recombinant transferment is allowed to grow on ampicillin and tetracycline containing medium, then also these can survive. So when in the ampicillin and ampicillin tetracycline media and tetracycline medium, if these colonies can survive, we can say that those host cells didn't receive the recombinant DNA or the gene of interest, that they didn't receive. So look here, non-recombinant transferments resist to ampicillin, and also they can resist to both ampicillin and tetracycline antibiotics, and also they can survive in a tetracycline medium. Now let us take a recombinant transferment. When the recombinant transferment is transferred to tetracycline containing medium, then it cannot survive, because here the tetracycline resistant gene is disturbed by the DNA sequence which we inserted into it. So these cells cannot resist to the antibiotic tetracycline. In the same way, if we transfer these colonies to the ampicillin and tetracycline containing medium, then also they cannot survive, because these can show resistance to the ampicillin antibiotic, because here ampicillin resistant gene is not damaged. But tetracycline resistant gene is disturbed, so it cannot withstand to the action of the antibiotic tetracycline, so those host cells will die. So when recombinant transferments are transferred to the tetracycline and ampicillin tetracycline containing media, they cannot survive. When this recombinant transferment is transferred to the ampicillin medium, it can survive, because in the recombinant transferment, ampicillin resisting gene is not damaged, so it can produce the product which can act against to the ampicillin and can survive in that medium. See here, when the recombinant transferments are transferred to the ampicillin medium, they can show resistance to ampicillin, so they can survive. When they are transferred to the tetracycline, these become sensitive to tetracycline, they cannot survive. But when these recombinant transferments are transferred to ampicillin tetracycline medium, then also they cannot survive. The reason is these are sensitive to the tetracycline, and even though they are resistant to ampicillin, they cannot survive because these are sensitive to the tetracycline. In this way, this selection is done by antibiotic resistant markers. This is called two-step selection.